Good morning, everyone. My name is Nabila Berliani Satyarahma Putri, and I am a student from Semarang State University with NIM 053. Today, I will present our laboratory observation titled Microscopic Analysis of the Cell Reproduction of Allium Cepa root. The study focused on visualizing cell division stage in onion root tips. Okay, mitosis is a fundamental biological process that drives plant growth, especially in meristematic tissues such as root tips. We chose Allium cepa or onion because it's easy to cultivate and its chromosomes are large and clearly visible under the microscope. This makes it classy model for studying the stage of cell division. There's a research objective. The objective of our study are first to identify and classify the stage of mitosis microscopical. Second, to calculate the mitotic index as an indicator of active cell division. And third, to observe how anatomical context is preserved in a longitudinal section. There's a methodology. Methodology, we use onion bulbs, vanillin hazel, acetylcarmine stain, slide, and light microscope. The root tips were stained, had gently cut, squashed under cover slip, and then observed under the microscope. At least 100 cells were counted per slide, and number of cells in each methodic phase was recorded. There's a methodic phase. Methodic phase consists of 5 main stages. And the one phase, there's an interphase. When the cell prepares for division by replicating DNA. And next phase, there's a prophase. Longest visible methodic phase, chromosome condense and become visible. Next phase, there's a metaphase. Chromosome align in the center of the cell. There's next phase, there's a anaphase. Sister chromatics paired and move to opposite poles. Last phase, there's a telophase. Nuclear membrane reform and the cell repair to divide. This state can be observed clearly using appropriate staining. This is observation result. And there's a table distribution of cell by methodic phase. Interphase, this percentage 56.2%, prophase 25.7%, metaphase 8.5%, anaphase 5.2%, telophase 4.4%. The methodic index was calculated at 24.5%, which indicates that the root tissues were actively dividing. There's a key insight. Prophase was the most frequently phase, which is expert because it lasts longer than other phase. Longitudinal sectioning allowed us to see not only the methodic state but also the anatomical zone, like meristematic and elongation region. No chromosomal abnormalities we found, indicating the method is safe and reliable. This technique can help students better understand plant growth at the cellular level. The last part, there's a conclusion. In conclusion, Allium Cepa growth is a great model for methods observation. The staining and discretion method would effective in handling cell division state. Longitudinal section at value by preserving tissues orientation. This laboratory activity support both biological research and educational learning. Okay, thank you very much for your attention. I'm open to any question and suggestion.